Welcome to Hephzibah's Simple Class. Today, I will be talking on regulation of Arabinose Operon and catabolite repression. In our previous class, I talked about an operon and described Arabinose Operon. I said an operon is a unit of genes which are under the control of a single promoter where RNA polymerase binds. An Arabinose operon or ARA operon is a unit of genes that encode enzymes responsible for the catabolism of L-arabinose sugar to D-xylulose 5-phosphate, an intermediate of the pentose phosphate pathway. However, on the Arabinose operon, the structural genes are togetherly transcribed into a polycistronic messenger RNA strand or split to create monocistronic messenger RNA strands that are translated separately into proteins. Moreover, Arabinose operon is regulated in the following ways. Negative regulation. During the absence of arabinose, though presence of glucose, bacterial cells such as Escherichia coli do not need the enzymes responsible for arabinose catabolism. Thus, the absence of arabinose allows regulatory gene product, also known as a dimer protein with two binding domains, to interact with ARA-I1 and ARA-O2 on the ARA operon, which subsequently leads to the formation of a DNA loop, that is, a curve shape, which in turn prevents RNA polymerase from binding to the structural gene's promoter, and therefore transcription of the structural genes is inhibited. Positive regulation. During the presence of arabinose and absence of glucose, the regulatory gene product acts as an activator, in the sense that arabinose binds to the dimerization domain of this regulatory gene product, changing its conformation and forming a complex known as regulatory gene product arabinose complex. This complex makes the regulatory gene product to fall off from the operator, ARA-O2, and also binds to ARA-I1 and ARA-I2. This thus allows RNA polymerase to bind to the structural gene's promoter, and therefore transcription of the structural genes occur. Nevertheless, activation or transcription of the ARA operon depends on the presence or concentration of cyclic AMP in the presence of arabinose but absence of glucose. The cyclic AMP beats up a complex with catabolite activator protein known as catabolite activator protein cyclic AMP complex. In the absence of glucose, catabolite activator protein cyclic AMP complex binds to the catabolite activator protein binding site. The catabolite activator protein binding site is situated between the control sites. However, the binding of catabolite activator protein cyclic AMP complex is responsible for breaking the DNA loop between ARA-I1 and ARA-O2, thus allowing the regulatory gene product to bind to ARA-I2. This thus allows RNA polymerase to bind to the structural gene's promoter, and therefore transcription of the structural genes occur. Autoregulation of the regulatory gene products. The expression of regulatory gene is negatively regulated by its own regulatory gene product. A high concentration of the regulatory gene product binds to the operator ARA-O1 of the regulatory gene. This in turn blocks RNA polymerase from interacting with the regulatory gene promoter. In another word, this simply means a high concentration of regulatory gene products inhibits its own expression. Now, let's briefly see what catabolite repression is. Catabolite repression describes the reversible effect by which during a rapid catabolism of one carbon source, uptake and catabolism of poorer substrate as another source of carbon is prevented. Also, catabolite repression is a system of gene control in some bacterial operons, in which glucose is utilized preferentially and the metabolism of other sugars is repressed due to the presence of glucose. In conclusion, catabolite repression in Arabinose Operon is a delayed response that directly controls or inhibits transcription of the structural genes involved in arabinose catabolism due to the presence of glucose. Here I come to the end of this class. I want to acknowledge and appreciate authors and owners of every material and image used in this class. Thank you.